Hello, students. Today, I would like to talk about a surgical technique called veno-venous ECMO and its consequential inflammatory reaction in a large animal model, more specifically in Vietnamese mini pigs. Let me first explain what veno-venous ECMO is. Veno-venous ECMO, or simply ECMO, which is short for extracorporeal membrane oxygenation, is widely used in a clinical setting. You've got a patient, and one day she wakes up and has difficulty in breathing. Naturally, she's taken to a hospital, and the doctors give her oxygen through a face mask. When this is not enough, she's put on a ventilator. In certain cases, however, it seems her lungs can't take up enough oxygen, and then an outside source of oxygen is needed. This is when doctors put her on an ECMO device. In COVID-19 patient care, doctors often have to resort to using veno-venous ECMO. As we know, in severe cases, the lungs are so damaged that even a ventilator is simply not enough to supply the body with enough oxygen. With such a temporary loss of breathing capacity, ECMO is an ideal choice for treatment. Unfortunately, one of the drawbacks to this method is that ECMO patients require monitorization at all times, and due to the shortage of staff at hospitals, it is impossible to handle too many devices in the same time in time of a pandemic. Now we know when veno-venous ECMO is used, so let's take a look at what it really looks like in the practice. Here you can see the venous blood exiting the body with the help of a pump. It goes through an oxygenator and is conducted back into the body. This way, oxygenization is put outside of the body while the blood's basic function of supplying oxygen to the tissue remains intact. In this video, you can see a tube containing blood from a vein and another with oxygenated blood in it. The venous blood comes from the person, goes through the machine, comes out oxygenated, and is diverted back into the body. There are several advantages to this therapy, such as supplying oxygen to the body while resting the lungs and damaging them much less than a ventilator. However, there are a number of disadvantages as well. As the blood comes contact with synthetic material and as the lung is excluded for the circulation, a large amount of heparin is needed to keep the blood flow clot-free. These disadvantages can lead to complications like irregularities in blood clotting, thrombosis, and bacterial pneumonia. But the most crucial complication is inflammation, whose significance I need to elaborate on more. Inflammation happens when the blood comes into contact with synthetic material like plastic. It induces inflammatory cytokine and factor eruption, which eventually leads to systemic inflammatory response. The rate at which inflammation happens in a patient is an important recovery factor. Lowering the rate of inflammation would mean a shorter recovery time, beneficial for both the patient and the hospital. Very briefly, let me draw your attention to the connection between the kidneys and an ECMO-induced inflammation. It always entails a renal macro and microcirculatory dysfunction due to the systemic inflammatory response. Now it's time I said something about what we aim to achieve in our laboratory at the Biological Research Institute. In our research, our main goal was to develop a clinically relevant veno-venous ECMO large animal model and to analyze the main components of the adverse reaction, mainly focusing on the kidney's function and the mitochondrial functions in the kidney. We put our experimental animals into two groups. One of them was the veno-venous ECMO group and the other was the control group. In the ECMO group, the animals were subjected to a 24-hour ECMO treatment, which was followed by a 6-hour post-ECMO period. We analyzed blood samples, measured hemodynamics and diuresis hourly. After concluding the experiment, we took tissue samples from the kidneys to measure mitochondrial functions and to run further biochemical tests. Furthermore, the asterisks indicate figures derived from measuring free radicals and counting white blood cells. Such data are direct indicators of inflammation in the body. In the picture, you can see how blood gas analysis is conducted. It's a technique that helps to monitor the physiological parameters of the animal in order to make sure it is in a stable condition. We use the Pico machine to measure hemodynamical parameters, for example, heart rate and blood pressure. The animal is under anesthesia, so the dosage of anesthetic and painkiller drugs was important as well. We also administered fluid to the animals to avoid dehydration, and we measured renal artery flow to see how much blood was distributed to the kidneys by the circulation. In this video, we took a blood sample during the intervention, and this sample is used in several ways. Firstly, we analyzed this, as I mentioned before, for monitoring physiological parameters. Furthermore, we prepared serum samples for later biochemical tests, and we measured its free radical content to assess intensity of the inflammation in the animal. Now let's turn our attention to the mitochondria. You must have heard the phrase that the mitochondria is the powerhouse of the cell. This is certainly true in normal physiological conditions. In the picture, you can see structure of the functional part of the organellum, and these blue bumps are called complexes. These complexes are key components of the action called oxidative phosphorylation, which produces heat and energy for the cell. What we see during ECMO is a mitochondrial dysfunction due to inflammation, oxidative and nitrosative stress, and ischemia or reperfusion injury. Because of this, the cell doesn't have sufficient energy, which leads to tissue and organ dysfunction in the end. We were mainly interested in finding out where the problem lay in the transport mechanism. In order to find about it, we use this machine, which is called a high-resolution fluorespirometer. For the experiments, we used kidney homogenate, and we mainly focused on complex 1 and complex 2 linked oxidative phosphorylation capacity. As you can see, the baseline and the complex 2 don't have significant difference in the two groups. However, the complex 1 was significantly lower in the ECMO group. This is good news because it provides an explanation about the renal failure we saw in the animals. Now let's take a look at the macrocirculatory aspect of the experiment. We measured the renal artery flow and the diuresis hourly to assess the kidney functions. We found that in the ECMO group, both the renal artery flow and the hourly diuresis were lower than in the control group, which means disorderly kidney function in the animal. And to sum our results up, we saw that in the venovenous ECMO group, the renal artery flow and the hourly diuresis were lower than in the control group. And on a cell level, we saw that the complex 1 had lower capacity of oxidative phosphorylation. However, the baseline respiration and the complex 2 linked oxidative phosphorylation didn't show any difference between the two groups. To sum our results up, we successfully set up and maintained a clinically relevant large animal ECMO model, and we proved the defect in mitochondrial functions in the kidney. This may play a key role in renal dysfunction. Thank you very much for your attention.